My name is Elizabeth Edwards, and I'm a professor in chemical engineering and applied chemistry at the University of Toronto. So this is actually a sample of moose rumen. So the insides of the guts of a moose. And since moose eat grasses, we thought that the enzymes that might be in their guts might be, or in the organisms that might be in their guts, might harbor enzymes, which are things that break down molecules. It can break down grasses. And if you can break down the components of grass, you can perhaps use that to make biofuels or to make other value-added compounds or to make methane. So in these bottles, they're kind of pressure, they can hold pressure. So if it decomposes to methane, the methane gas is within there. There's a rubber stopper. You can measure that gas with a syringe. And so basically it's what happens inside your gut, too, that causes gas. Happens inside a moose as well. But they're eating cellulose. They're eating grasses and things that we might want to use in a bioeconomy. So that brings us to the project that we're running. Okay. So bioproducts and enzymes from environmental metagenomes. And what does that mean? It just means that we're looking for new bioproducts that could also be microbial cultures. I work particularly in bioremediation, so we're looking at bacteria that break down contaminants. And also enzymes, because all of these bacteria harbor all these enzymes that are actually doing all the work, chopping up molecules and making them into new molecules. And so we've got a program that's screening enzymes broadly, and the genomics part of it, metagenomes, is just a fancy word for environmental genome. It's like this is a sample of groundwater and soil, or contaminated soil from a contaminated site in Ontario, and a metagenome would be taking that bit of soil and water and extracting all the DNA from there and looking at all of the organisms and their genomes in that soil. And that enables us to say, well, what can this, what if we know that if I put in here dry cleaning solvent in the top and it goes away, it's degraded. So there's some organism in here that's doing this, and then we have a tools by which to try and understand how that's happening. And that's the technology that we actually launched this project on, which was, I'd worked with a consulting company, and that's our end user, one of our end users anyway, is Geosyntec Consultants, and a spin-off company that they formed called Serum in Guelph that actually markets a culture that degrades dry cleaning solvents, and trichloroethene, which is the most common industrial degreasing solvent, is found in all industrial sites, and it's a very persistent carcinogenic compound or toxic compound in groundwater. And several years ago, we put dirt in a bottle just like this and squirted in some, dry cleaning solvent. And I came back 2 weeks later, and it was gone, and it was all converted to ethene, which is the non-chlorinated. So you had trichloroethene gone to just ethene, and all the chlorines had gone off to form HCl or salt in the bottle. And I couldn't believe it. And my mom had this red pickup truck that was really zippy, and she called it the kick butt truck. So we called it the kick butt culture, or KB for short, or KB1. And now they sell it commercially in Guelph. They grow thousands of liters of this culture to inject into contaminated soils and aquifer material in subsurface to degrade chlorinated solvents. And it's a great business. So we wanted to try and do that again, find sources where the bacteria had naturally become acclimated to whatever. So this was a contaminated site that had, for 50 years been exposed to these chlorinated solvents in the groundwater. So the bacteria adapt, right? They divide and multiply and change their genetics a little bit so that they can handle the toxicity of the chemicals. And so similarly to the moose rumen idea, it's that, the bacteria inside a moose are used to the stuff that the moose eats. And that's what they're acclimated to over long, long periods of time, evolutionary time. And therefore, they might harbor really interesting enzymes that we haven't looked at before. And all these genomic tools allow us to get at these enzymes really quickly. And then this kind of test, the idea would be that the genomics is incredible. It's giving us sequences to so many things that we have no clue what they do. And so how does one screen rapidly for functionally, enzymes that have a great function? So we're trying to develop an assay that's more relevant to real substrates. So this is just little pieces of ground up wood that have been run in 96 well plates, which is just, under a format where you can do 96 at once in little wells, tiny little wells. And then you add enzymes to these and let it incubate to see if it'll degrade any of the wood. break down the wood, which is what we're trying to do to extract the value out of cellulosic biomass. And then once it's finished reacting, put it on the Scotch tape, put it into a fancy mass spectrometer, and analyze the wood, the native wood, to see what's been degraded. And that's actually working quite well to screen. So we might find enzymes in here that degrade wood or wood-like products because they came from an animal that eats woody products. But then you need to not. This is still hard to analyze. You need to get down to what is the actual enzyme that's doing the job, or enzymes, or mixtures of enzymes. So that's basically the whole project. And the Genome Canada money has spawned something we've developed at the university. So I'm a professor in chemical engineering at the University of Toronto. And what we're creating at the University of Toronto is a Center for Applied Bioscience and Bioengineering in the engineering faculty, which is bringing scientists, enzymologists, and people that would normally be in the medical fields or biology or something right there, as well as people involved in sort of policy analysis, things like that, right in one center. And we got some funding from CFI, from the Canadian Foundation for Innovation, also from the Ministry of Research and Innovation in Ontario, to build an extra floor on the building to bring all these people and house them. Because I think the best thing to get innovation is to be chatting, chatting at lunch, students, working elbow to elbow. So that's what Biozone is, and it's leveraged. The Genome Canada stuff enabled us to. Both kind of happened at the same time, but they're leveraging off each other, and we're creating infrastructure, and then this is the research funding.